Okay, so ngarang dagi koi see dark reaction hogram neba. We were talking about Kelvin cycle and how glucose gets prepared during Kelvin cycle. Nainagri natra. Kelvin cycle ki matamda glucose sungbase. To prepare one glucose, how many rounds of Kelvin cycle needed? 6 rounds. And how many ATPs? 18 and 12 NADPs to darkar oikrabane. Hamain to na koi Kelvin cycle khangre ko. Kay Kelvin cycle si dark reaction ko ugrise. Because this particular reaction is independent of light. Independent in the sense that it is not directly initiated by the light. Light na initiate the reaction na TMAD. Hoy, light reaction gi product to the budi depend to wiko. Tudubu, light the directly depend to reaction di nati. Kay biosynthetic phase ko gridi because Kelvin cycle gi matamda glucose synthesized to wagi. Yare ko? Now, adu mayam se khangre ngasi sir na harak question asine muna taw ko. So let me just recall the small thing about dark reaction. The second step of photosynthesis, dealing with glucose preparation eva dark reactions. Sidagini ko khangba hogdose muna tasiko. Sigi dark reaction se ngarang aikoi na wari sanamina giba matamda dark reaction gi ihan hanba production first stable production kanane hena tamgiwa. The first stable product during dark reaction C, it's a how many carbon compound? It's a 3 carbon compound called 3-phosphoglyceric acid ni hena tamgi. Chumiko 3-phosphoglyceric acid, which is a 3 carbon compound. Ana ihan anba product to 3 carbon compound oigibagi dark reaction se kaisu kaugibana. C3 cycle su kaugibane. Chumiko ihan ana kanana explain to wa Kelvin explain to by Kelvin cycle su kaugi. Yareko, Hosik sir amuka su haigeko. In majority of the plants, the first stable product is a 3-phosphoglyceric acid. However, in some plants, so I'm using the term in some plants. Panbi kharada, not yamna yamni nataba. Panbi kharada. In some plants, the first stable production. In some plant, first stable production. Wait a second. In some plants, like kanada, like maize kumba, sugarcane gumba, sorghum gumba da check tawruba matamda ko. First stable production, 3-phosphoglyceric acid oil the one. Yani ko, in some plants like maize, as written in your NCRT, sugarcane, ay sugarcane si NCRT da iba ude, kangjan biro, and kana sorghum. Hariba Panbi Singh Shidadi, when we do check the first stable product, it was found that the first stable product is a 4 carbon compound. Or earlier, but name si Panbi Shidadi. Yareko, Yahanan ba stable compounds first stable product in some plants like Shidadi, first stable compounds. The first stable product was found to be a 4 carbon compound, and that 4 carbon compound is none other than, look here, oxaloacetic acid. Oiler में carbon क्या compound है बा 4 carbon compound. Dark reaction the dark reaction को because this part is also dealing with glucose preparation. Only thing is that in this special type of dark reaction, the first stable product is 4 carbon instead of 3 carbon. Hence, this mechanism, this special mechanism of dark reaction is called C4 cycle. 하이나 수강래요. 야래거 C kay koeba C4 cycle. So today we'll be talking about C4 cycle gi wari neba sana dois. 야래거 아도 코이나 강가다바세 C4 cycle 수동 kaine dark reaction. What is dark reaction? It is the second step of photosynthesis dealing with glucose preparation. And glucose adum prepared togani, the only difference is in C4 cycle, the first stable product is oxaloacetic acid instead of 3-phosphoglyceric acid, which is very common in many plants, right? And apoy na hujik tam doshidi common native. Yareko, we'll be talking about some special plants, kharaki wari ne. Hairiba panbi se, karamba panbi. The plants like maize, sugarcane, and sorghum, where the first stable product is oxaloacetic acid, or in other words, C4 cycle chathava panbi singse, we call it C4 plants. Kay koiba mo C4 plants, kawi and haido. Yare ko, adudi ngasi tak to ise kaineba, ngasi taktoisi will start with C4 cycle taktane. Shall we start? Yareko? Okay, can I change the screen? Look here. Ngasi taktoisi muna tabiu C4 cycle. C4 cycle was for the first time explained by two scientists, one called Hertz and another called Schleck. Hence, it is also called Hertz and Schleck cycles. It is also called Hertz and Schleck cycle. Why? Because it was first explained by Hertz and Schleck. I was scientist Anish explained from Why called Kelvin cycle? Because the mechanism was first explained by Kelvin. Yareko? Adumaina? Aduna ngashigi topic sina koi C4 cycle tamdane. Yareko? Aduna kyam hodringaida akoina kangdoise C4 cycle sidadi the first stable production kananeva the first stable compound formed during C4 cycle is a four carbon compound called kana oxaloacetic acid neva. Carbon kya compound neva? It's a four carbon compound. Yareko? And the first carbon dioxide acceptor. What was the first carbon dioxide acceptor in C3 cycle? RUBP, which is a five carbon compound, right? However, here the first carbon dioxide acceptor is a three carbon compound. C4 cycle the iyan carbon dioxide acceptor se kananeba three carbon compound called phosphoenol pyruvic acid or pyruvate hairse. Yareko? When nungai full oina thage phosphoenol pyruvic acid. ID acid ki form do ina dum haige ko sum simaina nungai haibana. Pyruvic acid noina nungai jadre hargana pyruvate oina hairasu yai. Yareko? It's a how many carbon compound? It's a three carbon compound. Yareko? Aduna kem takpa hodringaida simadi hana khamina tare. C4 cycle nadi ahanba production oxaloacetic acid ni aduga ahanba carbon dioxide accepted na phosphoenol pyruvic acid ni haibasima. Yareko? Hairiba C4 cycle se masi kanada chatheva. It occurs in C4 cycle occurs in group of plants called kaikoba C4 plants. C4 plants the chatheva. Yari bromium, C4 plants. May I know some C4 plants mentioned in the NCRT? I just wrote in the previous slide. They are maize, sugarcane, and sorghum. Sugarcane and sorghum. Yari ko mayam. Now, huzik sir na haraktoisa C4 plants karamba makhalgi panvino haitardi C4 plants are the plants that are normally grow in kai jagada hoge moise haibakandana dry tropical areas tawe. Tropical akoi biodiversity and its conservation ta kidangairi tropical areas kaidale equator mapawaida saramgadra so they are having high temperature zone. Yani tropical rainforest, Amazon rainforest, tropical. Adi uayam horam ni oxygen satina concentration wangi. Yani adi oxygen yamna leiba mapam, temperature yamna wama mapam, light na directly perpendicular yai jagane equator di. Light intensity yamna wangba jagane. So there are high chances of photorespiration. Yani adudi aina hairaktoise me hairiba sorghum hairiba sugarcane hairiba C4 plants normally kadaiwaida hauba jibano panbino haitari dry tropical areas sing the dry tropical areas or forests I should say kadaiwaida high temperature area the hauba yani high light intensity leiba mapa sing the hauba moise karamba panbigi warina C4 plando. Adwai taragadi aishu khali noisu khalamganiba. They may face the problem of photorespiration mani haidana because we discussed that at high temperature, at high oxygen concentration, and at high light intensity, Ruvisco behaves as oxygenous when a behave the way haidana. Adubu C4 plants though they are growing in this condition, they do not have photorespiration later. Yarniko, pain adum photosynthesis rates wang na tawe. Kaigino haina check tawrakanda C4 plants have special adaptation. Okay? Moi the nature sinane ꯏꯁꯣꯔꯁꯤꯅ ꯁꯦꯝꯕꯤꯔꯛꯄ ꯀꯥꯟꯗ ꯃꯣꯏꯒꯤ ꯑꯦꯅꯥꯇꯣꯃꯤꯗ ꯈꯔ ꯆꯦꯟꯖꯦꯁ ꯈꯔ ꯂꯩꯔꯝꯃꯦꯕ ꯌꯥꯔꯅꯤꯀꯣ C for plants have special adaptation to avoid the problem of photorespiration photorespiration dagi ngakthokjaba yanaba moida special mechanism khara yawe okay so we'll be talking about those adaptation do han tamgini isini sarna taise yare ko chumnagi normal plants so itari hairiba condition sidari compulsory kai chatharane photorespiration chatharadamneba adubu C4 plants dadi photorespiration chathatrise kaigino haitaragadi moidadi kai yaweba some special changes are given to the plants so as to deal with this problem okay so we'll be talking about those adaptations found in C4 plants hana tambatare so we jump to the adaptation yarniko ana ai adaptation se asum saito ine ige munata se ko so hojik tak to se adaptations found in C4 plants. Adaptations found in C4 plants. C4 plants the phangba adaptation khara naina si. What adaptation? Adaptation used to prevent photorespiration dagi ngakthok chanaba yawa adaptation haitare in C4 plants. Yare ko? Let's see the adaptations. Kari kari adaptation yawi amuktang yengminase ba sir. Yare ko? C4 plants singda photorespiration dagi ngakthokjanabagidamagta the nature has provided to the plants two adaptation yareko adaptation ani khak thamami adaptation ani se kari kari adaptation no haiba kanda number 1 all C4 plants they show a special anatomy called as Kranz anatomy hena ama yawe yareko I will explain just wait one is called kayaba Kranz anatomy we'll discuss Kranz anatomy in a minute Kranz anatomy haibama and all C4 plants show dimorphic chloroplasts. Chloroplasts makal ani yawe moida. Yare ko dimorphic chloroplasts. Aikoibu lip ki anatomy ningsang nariko mea. Lip ki anatomy. Lip has got two surface, upper epidermis and lower epidermis. In between is filled with mesophyll cells na filled away. Haigi. Aduna sardong haktang sigi wari se lakpa yanaba lip to yeke to banate just anatomy dum nang machin amtang ningtang se si lip ni lip ki upper surface is also called adaxial or abaxial lip arni ko upper surface is covered by upper epidermis ningsangi ko maya likewise lower surface is also covered by lower epidermis lower surface is called abaxial epidermis or abaxial surface sign ko yare ko Horan veins yawa jagaduda kailai we have the vascular tissues, right? The vascular tissues like xylem, phloem, leidoni. And these vascular tissues are surrounded by a layer. Cell layer, we call it bundle seed cell. Kawi haigi. Chumbra? Okay. In between, the upper epidermis and lower epidermis are filled with cells called as mesophyll. In dicot, those mesophylls are differentiated into two layers, upper and lower. The upper are tightly packed parallel to one another. We call it palisade mesophyll, while the lower are loosely arranged with large intercellular spaces called as spongy mesophyll or spongy parenchyma. There is no such differentiation in monocotonousy differentiation like there. Yareko mea. So this is the normal anatomy of lip. Lip anatomy se asine. Yareko. Adoi tari. What is this? Mesophyll. Okay, pellicet spongy harakanova. Simply mesophyll. Mesophyll. Mesophyll lies towards the upper and the lower side. The upper side lomda laibanina they will be directly exposed to light, right? Saramgadra dudi. Saramgani moise. What is this cell? These are bundle seed cells ne. Chumiko manung lomda laibra. Saram moy dudi. Lakpa kay do mapan gi mesophyll cell duna ngakto moidro. Ana bundle seed cells ne ingam gani. Yare ko. Aduna hozik sir na hairak to isa. Mathaki mesophyll cells ita saringai sida mesophyll cells ina rubisco enzyme hapu tari kai loksal ania. Oxygen loksal oidra sabanina. Ana nature sina khangdana mesophyll cell da rubisco yohalam dawan. Rubisco se kadai wai doina hapamba khara ingba kai wai da bundle seed cell doina yaudone. Yare ko. Aduna I will come to the slide. Look here. Dimorphic chloroplast hairadine. C for plants in the chloroplast makal ani phangeba. Noi khangani yo. Chloroplast makal kya phangeba? They have got two chloroplasts. Kaiwaida, one found in the mesophyll and another found in the bundle seed cell. Amana mesophyll they have chloroplasma, amana bundle seed cell they have chloroplasma. Sumain to gai ge line aba. Yare kwa, amana kai da phangba mesophyll cell da phangba chloroplast. Mesophyll cell da phangba chloroplasma. Moishibu saramdoro ingamdoro kaigino saise mathak thangbagi chumiko aduna sarabanina moidadi kai hapam da wano nature sina rubis ko apsal da otherwise kai problem lebe ai photo respiration thoki batare haina asaba mafam dai rubis ko lokthwe yare ko amana kadai waida phangba chloroplast bundle seed cell da phangba chloroplast bundle sheet cell chloroplast bundle seed cell da phangba chloroplast aduna chloroplast se makhal ani khaidoeba यारेको मैम mesophyll cell da ladybug chloroplast se sidhi kayaudega there is no rubisco sidai rubisco enzyme yaudeba rubisco yaude yarnika rubisco gi muhuttane enzyme amoina hapameba that enzyme is called pepco or pepcase haina khangne ai hana hairake ko enzyme se kai koiba PEPCO phosphoenol pyruvate carboxylase haina carboxylase di kai khaloxin hamane so no matter what happens in the environment sagera ingera oxygen wangera nemgera light intensity wangera pepco is only capable of fixing so aduna phaniyam sajarasu carbon dioxide lotion bangamba enzyme khaktang moina hapam bane kai waida mesophyll cell whereas in bundle seed cell remember is it in the asaba jagara ingba mafam la adi there is no problem of having rubisco right and sida waina rubisco yamba rubisco present sida waina hapam meba rubisco se rubisco present ngahaler gyan tarakini kitang muna tamina gisiko sibo yari bro mayam sumain todana chloroplast makhalani phangeb one chloroplast having pepco another having rubisco yarniko sumaina chloroplast makhalani laibasi we call it dimorphic chloroplast haneko hangadro aduna even if the mesophyll cells are exposed to sunlight even if the mesophyll cells are exposed to high oxygen concentration they won't be able to uptake oxygen because mesophyll cells do not have rubisco yaudre kai wene yaure ana mapanda sajarasu mapanda oxygen concentration wangjaba tarasu kai wene loksalani carbon dioxide wene loksalani haidana one adaptation to prevent photorespiration. Let's talk about the second adaptation. The second adaptation is called kai Kranz anatomy. Let's see Kranz anatomy. Kranz anatomy say NCRT the iris sir kitang satisfied oidego. Look here, let's back to the diagram. This is upper epidermis, this is lower epidermis, right? What is this cell? Bundle seed cell. Bundle seed cell si rubisco yaubra. So anyhow akwai shahanba yade. Yani ko inghandragadi saharadi adum kai thone. Adunane kamain tawgira akwai ba cell sibu kai kauge. Mesophyll cells however. The mesophyll cells surrounding the bundle seed cells are arranged in the form of ring. Ringy form the arrange the one ida. Let me repeat again. What is this cell? The bundle seed cells, together with the mesophyll cells, are arranged in the form of ring. In other words, atopa londa amda haragana, the bundle seed cells is being protected by mesophyll cells from all the direction. So that even when there is light or heat, they get absorbed by the mesophyll cell. So that bundle seed cell remain cool. Todum ingalayani. Yareko. NCRT dana kamaini ige the Kranz anatomy sa. Bundle seed cells are arranged in the form of ring. Kana. Bundle seed cell se ring gi form the arrangement of the Kranz anatomy kawi na hayaba. Ado bundle seed cell khakta nateba ring da leitoise. Bundle seed cell do nateba main di. Bundle seed cell together with the mesophyll cells to neva. If arranged in the form of ring, we call it kaikoeba Kranz anatomy. Why the mesophyll cells surrounding the bundle seed cells should be arranged in the form of ring? Because their job is to protect the heat. Okay? So that bundle seed cell do kaidora lainaba inga laigadaw. Yareko, bundle seed cell are arranged in the form of ring hydrogen. Nahane ko nigla iba lip anatomy do loy ring way. Oidrane. ꯑꯣꯏ ꯑꯗꯨꯗꯤ ꯑꯗꯨꯁꯨ ꯀ꯭ꯔꯥꯟꯁ ꯑꯦꯅꯥꯇꯣꯃꯤ ꯇꯥꯔꯅꯤꯕ ꯑꯗꯨꯗꯤ ꯌꯥꯔꯅꯤꯀꯣ ꯑꯗꯨꯕꯨ C4 plants ꯀ ꯑꯇꯣꯞꯄ ꯄꯥꯝꯕꯤꯒ ꯈꯦꯠꯅꯕ ꯑꯃꯁꯨ ꯂꯩꯀꯣ ꯕꯟꯗꯜ ꯁꯤꯠ ꯁꯦꯜꯁ they have a very thick cell wall ꯊꯤ ꯊꯥꯏꯕ cell walls ꯌꯥꯔꯅꯤꯀꯣ cell wallꯗꯣ ꯊꯥ ꯊꯥ ꯂꯥꯎ ꯇꯥꯔꯗꯤ ꯀꯩ ꯆꯪꯉꯝꯃꯔꯣꯏ heat ꯆꯪꯗꯅꯕ ꯏꯪꯉ ꯂꯩꯅꯕ ꯌꯥꯔꯦꯀꯣ this arrangement is called Kranz anatomy. So I'm going to write Kranz haibase. Kranz means reed haiba. R-E-A-T-H. Reed adi kadai bu kawi nahamok su sarna hai gimni. Yani ko angel gi makukta phaba ring no. Yani ko reed means ring haibane. Yani ko kanano ring moina leido se. Ada ID NCRT dadi focus tawdoni sumaino oina haigeba. Lokthokhiya na. Bundle seed cells are arranged in the form of ring together with the mesophyll cells. Bundle seed cells are arranged in ring like manner along with kanagaloinana along with mesophyll galoinana arrange tawba taragadi masigumba arrangement si trans anatomy ko this anatomy also helps prevent the problem of photorespiration so C4 plants the temperature wangjarasu oxygen concentration wangjaba taragasu light intensity wangagasu photorespiration thokte kaigino na haltari we have two answer one it has got trans anatomy and second, it has got dimorphic chloroplast leibadani henakumdo. I hope you understood the part. Yarnikwa. Now, Hosic mechanism adaptations a khangabadina. Let's jump to the mechanism. What is C4 cycle? Horosira Ardra. Yarnikwa. Okay, look here. Mayam Yari bro. Yariko. Now look here. Let me start with C4 cycle. Hodromi the actual mechanism. Mechanism horokanda. What is the another name of C4 cycle, please? It is also called Hertz and Slack cycle. Hertz and Slack cycle. Okay. Now, amtanga recall tawinasi, what should be the first product form? A four carbon compound called oxaloacetic acid. The first carbon dioxide acceptor, three carbon compound called phosphoenol pyruvic acid. Yareko? Hangeko? Sigi part si ingathrage duri yarni yaradra mayam. Okay, so mine ingat may mata horen chan na mas mine ingat yengasi ko duri project mechanism horasi mechanism holdringai da sir cell ani khakte yakige mapan thangba cell du kaino mapan thangba cell du kaino mesophyll cell manung thangba cell du kaino bundle seed cell very good manung thangba cell du na bundle seed cell ne yariko mayam okay what is this cell this is mesophyll makhon thoktana nofet toba misu ui ai swai si kaineba mesophyll cell manung sina kai bundle seed cell ꯆꯠꯄ ꯂꯝꯕꯤꯗ ꯆꯠꯄ ꯀꯥꯟꯗꯒ ꯅꯣꯛꯐꯦꯠ ꯇꯧꯗ꯭ꯔꯣ ꯉꯁꯥꯏ ꯃꯥꯅ ꯑꯩꯉꯣꯟꯗ ꯃꯨꯟꯅ ꯌꯦꯡꯉꯛꯄꯗꯣ ꯀꯥꯏꯅ ꯁꯣꯝ ꯌꯦꯡꯉꯣ ꯉꯥꯏꯈꯣ ꯑꯗꯨꯏ ꯃꯇꯥꯡ ꯋꯥꯠꯂꯤ ꯃꯦꯗꯤꯀꯦꯜ ꯐꯥꯎꯔꯗꯤ ꯌꯥꯔꯛꯇ꯭ꯔꯕꯅꯦ ꯌꯦꯡꯉꯣ ꯁꯣꯝ ꯌꯦꯡꯉꯣ ꯐꯥꯒꯤꯗꯧꯅꯦ ꯁꯣꯝ ꯌꯦꯡꯉꯣ okay which one ꯁꯤꯁꯦ ꯀꯔꯝꯕꯅ ꯁꯥꯗꯣꯏꯅꯣ what enzyme is present here pepco no rubisco here right how about here so let me put in the mesophyll cell we discussed that pepco is there phosphoenol pyruvic acid what carboxylase ana pepcase sukoy ano NCRT dadi pepcase ano ina even ko pepco hairage sardi bundle seed cell na kayaweba we have rubisco what is rubisco capable of fixing it is capable of fixing carbon dioxide as well as oxygen. Though, however, bundle seed cell si kay daw ing bazagara kay win option niya. Okay. Something yung siko. Hosik se. Ay na lawri ba panbi se. ꯑꯩꯅ ꯂꯧꯔꯤꯕ ꯂꯤꯞꯇ ꯌꯥꯎꯕ cells ꯅꯦꯕ mesophyll cells and bundle seed cells are the cells of the leaves, right? ꯑꯩꯅ ꯂꯧꯔꯤꯕ leafꯁꯤ maize-ꯀꯤꯅꯦ ꯂꯧꯔꯁꯦ. This is a C4 plants. And surrounding the plants temperature ꯊꯤ ꯋꯥꯡꯈꯠꯂꯛꯑꯦ ꯂꯧꯔꯁꯦ ꯍꯧꯖꯤꯛ. ꯌꯥꯅꯤꯀꯣ? ꯌꯥꯝ ꯋꯥꯡꯉꯦ ꯍꯧꯖꯤꯛ temperature-ꯁꯤ. What problem might have happened? Photorespiration. ꯊꯣꯛꯏꯕꯗꯣ. ꯀꯩꯗꯧꯕꯒꯤ mesophyll cell-ꯗ ꯀꯩ ꯂꯧꯁꯤꯟꯕ ꯉꯝꯕ enzyme ꯌꯥꯎꯗꯦꯕ. oxygen ꯂꯧꯁꯤꯟꯕ ꯉꯝꯕ enzyme ꯌꯥꯎꯗꯦ. Temperature ꯋꯥꯡꯖ ꯇꯥꯔꯁꯨ. ꯑꯗꯨꯅ ꯀꯩ ꯑꯗꯨꯝ ꯆꯪꯉꯅꯤ? What enzyme will be coming in? What will be coming in? Carbon dioxide changeva. Though the temperature is high, carbon dioxide is coming in. Changeva carbon dioxide se. It is accepted by how many carbon compound? It is accepted by 3 carbon compound called phosphoenol pyruvic acid. How many carbon compound, please? 3 carbon. Please read out loud. Phospho. The phosphate ama yauriko. Mayam gyan tami yaure ka yawe se. And the first product form is oxaloacetic acid. How many carbon compound is this? 4 carbon compound. Since the first compound is a 4 carbon, hence the name C4 cycle is given. Then this reaction is catalyzed by pepcode, not by Rubisco. Very good. The reaction gets catalyzed by phosphoenol pyruvate to the kay hapsan mga enzyme. Carbon dioxide hapsan mga bagidamagtane pepco haina kaurise. Phosphoenol pyruvate carboxylase haise. Yari ba siba wada? Yari ko. Now, oxaloacetic acid se. In the next step, it changes into a 4 carbon compound again called malic acid. The change there. Kay carbon compound 4 carbon compound called malic acid. Is it a cell? Sisu cell ni ko. Cell ni si plant cell ngakta. Plant cell gets connected through plasmodesmata. Horin hone eba ni si. Yar ni ko. Ana mesophyll cell da lairamiba kana se. Now it moves into the bundle seed cell, which is a cooler zone, right? Kadai da kumdare hoze bundle seed cell. How? Through the plasmodesmata. NCRT the plasmodesmata level to one. How many carbon compound is this? This is a 4 carbon compound. Very good. Hosek bundle seed cell that kay yaw lere, malic acid yaw lere ne. In the bundle seed cell, the malic acid get disintegrated into a 3 carbon compound called pyruvic acid. The kaida pyruvic acid. How many carbon compound? 3 carbon compound. So 4 carbon change into 3 carbon. 1 carbon is lost here, right? That carbon is released in the form of carbon dioxide. Yareko sibo. Yareko, this carbon dioxide say, are there any carbon dioxide acceptor here? Yes. What enzyme? Rubisco. With the help of Rubisco, this carbon dioxide is added to RUBP, which is a how many carbon compound? It's a 5 carbon compound. And it forms 3-phosphoglyceric acid. How many carbon compound? 3 carbon compound. Then 3-phosphoglyceraldehyde. What is this cycle? So the Kelvin cycle si chathare. Yareko, amta tago ko kaino chathari se Kelvin cycle. Mayam yari bro. Ay adudi hairaki amko. Sir se aswai siga yana dev. Sir se Rubisco ne lor seva. Aswai siga yana drev. Ay aswai da chang mutare ay kai will lor tore. Aswai shem sayeva. Aduna ay aiga aswai siga yana dabanina yana ba mi ma wen thara carbon dioxide lohalakwan. Mesophyll cell ga Rubisco ga yana deva. Why? Because mesophyll cell is very hot. Hot adi noin khali hot tunati. Samemo. Yare ko. Yam saeba. Adi Rubisco se adu da thu tardi kai will loksalani. Adu tawdanaba Rubisco na mai mamana bo in thawan pepco ko. Pepco na kai lole ade. Pepco na carbon dioxide pusale kai tana aying batana. Pepco na pusala po carbon dioxide to aying mo phom den pithori. Sidana kana le me. Oh, yana ba magi through the will hola swide when a fix the one. Kambro, swai se kaino. So bundle sit cell. A swai shina mesophyll cell. Mesophyll cell the I chat lutar the problem talk to reva. Kai problem. So to avoid that photorespiration, I chataba. Yani ko aswai ga yana ba mi mo in time. Swai yana mi de min kai goeba. To pep ko na matang pusale swai ba dong carbon dioxide low one. Lower ba? Adu kay chathare. So I can say that Kelvin cycle is a part of the C4 cycle of C reaction sida. Here ko. What is formed here? Pyruvic acid. Kaida. Howzik bundle sit cell the ladiba pyruvic acid se. Now it moves up into the kaidam kakhleba. Sorry. It moves up into the mesophyll cell dam kakhleba. Kaida kakhleba. It moves up into the mesophyll cell. Pyruvic kai acid. How many carbon compound is this? Three carbon compound. And what is this? Phosphoenol pyruvic acid. Sina pyruvic acid ka. Adi pyruvic acid tagi phospho. Wala kedi phosphate हम half the DR ne. So in the reaction, we are going to add a phosphate. Sidagi CM olev. Yani ko. To do this, what are we going to add? We're going to add phosphate, right? Phosphate apadi nahanggay tamak pase. ATP sidagi phosphate option card it should form ATP, but unlike other reaction, it forms AMP form to it. Yani ko triphosphate ay monophosphate ulayra adi phosphate kya shali ATP dagi AMP on basa. It is similar to the use of two ATP shin gamani haine. I will show you here. Look here. Look here. See ATP ama sumay layrameba ko. ATP dagi kay ama satuoba. Inorganic phosphate am satari kay form to eba. ADP, right? Umaga reaction ge kuina chang na se copy to ge. Look here. In both the Shigi reaction ani shira how many total phosphate is removed? Phosphate ani log to remove two phosphate, how many ATPs consume of? To the ATP ani log thok pase, sorry, phosphate ani log thok pase, ATP ani shina yi haibaga mana yi. Aduna ATP dagi AMP on the means equivalent to the use of two ATP. Aduna akhoi line aba I adom sida 2 ATP haina irageba ka shukai de dana do 2 ATP na direct isan reaction ka kitang dikha nei ko mani haibo ani ana I adom sida kayolani 2 ATP haina waina irageba ka shukai da bro. So this is called C4 cycle. C4 cycle I say. Yani ko how many carbon dioxide is coming in? How many? Is one carbon dioxide enough to fix one glucose? Wait a second. Lagi kay sumorani glucose sungdoni ko ngarang tamgibaduni sidi ay tak trasu yarabani. Yare ko do one carbon dioxide enough to prepare one glucose? No, we need how many carbon dioxide? So we required 6 rounds of C4 cycle to prepare one glucose. Glucose ang prepare to the C4 cycle kya dakar hoeba? C4 cycle taruk dakar hoeba. Ay na uli sila kya na? Amma khatane ay na uli sila. I'm showing here only 1 round of C4 cycle. Now the question is, question ang age tabi ro ba? Landoy ni edom khangna to prepare, let me write here, to prepare one molecule of glucose. To prepare one molecule of 1 mole of glucose. Glucose molecule ama prepared tauge hai tari. What are the things that we require? How many carbon dioxide will be required? Will require 6 carbon dioxide required. Carbon dioxide taruk ama darkar oirni. Ereko, how many rounds of C4 cycle? 6 rounds of C4 cycles needed. Cycles needed. How many ATP? How many ATP will be needed? Okay, 12 na tira. Say mo si. Is Kelvin cycle a part of C4 cycle? Ari ngarang Kelvin cycle the round truk setup the ATP kyam changrage. Sida 18 ama sidagi 12 ama 18 plus 12 equal to 30 ATP neba. Hangro C4 round taruk lak chatha tari mai manung diawa Kelvin cycle su round taruk lak chatha mani. One carbon coming in, when one carbon dioxide coming in, 1 Kelvin cycle. 6 carbon dioxide coming in, 6 Kelvin cycle chathara moidro dudi. Kelvin cycle round tarukta kaya sungre have dudi ngarang khangre. How many ATP? 18. Sidai round namada ATP kya sangi shira. Round taruk ki trying to la ani shiragi. 18 plus 12 equal to, so we'll require 30 ATPs. ATP kya changeba? 30 for preparing one glucose molecule. How many NADPH2? Very good. Will require 12 NADPH2. Kanada ki lakpano sila. C3 cycle, Kelvin cycle dagi lakpano. Khangro mayam. Yardi ko. So these are the things that we needed for preparing 1-glucose through C4 cycle name. Understand tawre haina loba yarabra? Yarni ko. Sida I kanam haige trombane. Okay. Yarni. Yare ko. Ay si bo khangi han lor si khaktane C4 cycle da atopam te yaudi. Yare ko. Sida kai yaram tarde rubisco yaram adi kai from thokane. Kai gino swaise. Ham sabagi. Yarni ko adu thoktanaba enzyme se tongan yalam. Enzyme se cell manung de na te ko yaudi se. Kai cell na se cell di manung da chloroplast yaram moidra chloroplast adugi chloroplast mesophyll cell manung de yawa chloroplast da kai enzyme ina hapa. Pep ko oina. Sigi bundle si cell gi manung de yawa chloroplast na kai oina hapa. Adi chloroplast ani do manadi hayai. One is having pepco, another is having. That's why I'm saying dimorphic chloroplast. Hai do nani. Yarage. Yareko. Any doubt eva hangwayani? Can I change this slide? So let's talk about the last topic of the chapter, that is factors affecting photosynthesis. Seeing us. Yung sila? Shidai question tiya hangbani ko. Shidai factors shidai. Ana ay ita oil. I'll be talking only about carbon dioxide and temperature. I'll focus on that part. Light part, haipetang di hai ni paga. Nangagadi pasi. Nangbudi nangani. Ado explanation di am sa factors affecting photosynthesis. Tested cocktail kanguk phenol maringda pudoniko. We'll go there. Thrown by nam done ko. Hagi tomni sumbu buroi. There were factors affecting photosynthesis. Listen here. Photosynthesis है करम factor the depend तौगे सिने यानी का photosynthesis से कदाई the depend तौगे हाइबा कांदा NCERT the पीबे information की मतुंगिना मकोई photosynthesis affect तो factors है we can divide into two. One is internal factors and another is external factors sign ani thong kai pe one is kai amana internal factors, one is internal factors. Internal factors are the one controlled by the gene. Gene na sanarak pa singsawarak ay hierarchy. And external factors. Light yam na itradi kay daw kay photosynthesis rate kay daw kanya. Handaroidra na handarakani light hay say external factors ne. Carbon dioxide already photosynthesis to hai that is external factor. Water yaw the yaro. Yani ko. Congressman I say okay, lip pick panga lip achawonga photosynthesis kanan to ne. Achawona, the broader the leaf, the more the mesophyll cells, the more the mesophyll cells, the more the chloroplast, right? Ado lip sets over pick by it's again controlled by the gene internally. Aduna mo internal factors sa lakani. Yare ko, aduna internal factors sida kana chinge haven't when I'm saying internal factors, it include the size, number, masing mana pung na pan ba hinaw duga mana kitang pan ba hinaw duga pung na pan duga photosynthesis hen batabani natrane number orientation sunlight on the exposed or leba mana duga sunlight on the gi on the leba duga karam bana exposed to ne photosynthesis to tamni natra orientation pan bagi mong orientation NCRT hangdok tardi atopa ka yauri game view yawam thi khang drama le haying na paikana si punsigi ahan baratai si Hyrodri. One more, amo waliba. Yes, chloroplast. Matang the mesophyll cells hybrid chloroplast. Chloroplast adi sinida. Lip amagi manunga. Lip anile rameva. Lip amada na chloroplast tara yawa. Lip amada chloroplast kun yawa. Maduri kun yawa na photosynthesis to bam thokra ne. Chloroplast, mesophyll cells. Mesophyll cell masing dasu depend to gadra. Tawni mesophyll cells. Yani ko. So what I mean to say is that the size of the leaf, the number and orientation of the chloroplast mesophyll cells will control the photosynthesis. Okay? Yani mesophyll cell masing yam yamaba, chloroplast masing yam yamaba, orientation se fazana light lom do onjala leraba, more photosynthesis. Yani chloroplast yam yamamdraba, mesophyll cell masing yamamdraba, orientation si light lom de low la leramaba, low rate of photosynthesis. Yani and say lipstick yam sauni. Yani chloroplast masing na kyam yauni, mesophyll cell masing na kyam yam yaw do noise. It is again controlled by genes, the control tail. So these are, I can also write, these are genetic factors now. Yarabo simaw? Now, ako'y ngasi explain tawdosidi internal native. We'll focus on the external factors. Yani mapandagi darkar oiba. May I know some raw materials needed for photosynthesis, please? We require, let's see. Yani let me start with, look here. Let me start with light. Light ama darkar oy natrane. We require light. Uh, let me put water. Let me put temperature. Sidi kano nate ko ay na ise raw material nate ko. Factors affecting photosynthesis. Temperature na wangadi. Photosynthesis rate wangadra name gadra. C3 plants ko inadi temperature ya wangadi kay dograni. Photorespiration room do in a shift dograni, so rate of photosynthesis decline. However, C4 plants they respond better to higher temperature. Higher temperature to respond for the num tawe. Adu ka hena wangalori ta gini ko. Kay gino. Every reaction cease at high temperature. Why? Ceases. Because at high temperature, enzymes get denatured. Yani ko, ang hengatpam topat chang de hengalura budi animak taloy impact budi lay niba. But C3 plants na am na pamjaba temperature city about 20 to 25 degrees Celsius why shinel. Ado horen C4 plants kiri 30 to 40 degrees Celsius ne harakaniyo. Yare ko, ana ahom suba aina talk to I will be talking about temperature, and last not the least, we have the most important limiting factor ni na horen even ega pamina do NCRT, and that's none other than carbon dioxide. Yarabo, and my explanation will start from this carbon dioxide holdene. Yaradra, I explain to way agirka. I will first define a law law of limiting factor. Can I change the slide? Halaki ko mo. From side to explain to gise. Jose I na talk to say law of limiting factor. Hana taraba? Tare ko biology da khatara yoy se. Bio de yawe haibabu ngasi khatang khangja da ngai ri na kumo. Law of kai limiting factor. Yare ko. Moi like ta irido khara janjan oina isali ai khatang simplify toroke somde yengsi. Photosynthesis ta involved the raw materials kara carbon dioxide. Yes, very good. Water. Okay, I'm putting light. So pahira gumsi ohum sinayala si light. Very good. For example, hojik sir na low this som nam ting biroga. Photosynthesis the glucose preparation to wanine ka. Photosynthesis se to gai tari yamdra minimum requirements hai carbon dioxide taruk changi lor se. Ay taruk changbra changdabra khangde ko. I'm just assuming that for photosynthesis to take place, at least how much molecule of carbon dioxide is required? 6. And let's suppose about 10 molecules of water is needed. And let's suppose about how much? Like 50 units of light is needed. Yani ko 50 candela. Dakar way in lower Yareko, yam the minimum requirement ngakani ko adi 50 data thargadi photosynthesis to roi haini 6 data thargadi photosynthesis to roi haini 10 data thargadi photosynthesis to roi haini ba sir na ise gyan ta bro tari ko mayam okay tardi yung som dam tang hairake sir yam the carbon dioxide minimum carbon dioxide requirements se kya neba let's suppose 6 units of carbon dioxide is needed 10 units of water is needed dakar oire lorse Eureka! And 50 units of light is needed for photosynthesis to occur. Now, how's it now? Sir, na, mana darkar o is 10 units eh? Sisa, it's been increased to 1,000 units the hengatle. Hengatle ko hojik, and also the light say 50 khak laser today khara hen official. Let's suppose about 500 units the official lower se. Yani ko, but let the carbon dioxide be fixed at 6. Will the rate of photosynthesis be increased? Sini questions. Yani ko, Congress and I say, any reaction, so in a law of limiting factor, I say photosynthesis khattaginative. Pitrivi the top any reaction they apply to whatever law name. Sigi law sina kai higam tamda particular reaction amagi rate set. It will be decided by the factor which is present in the lowest quantity. ꯈ꯭ꯋꯥꯏꯗꯒꯤ ꯌꯥꯝꯅ ꯌꯥꯎꯗ꯭ꯔꯤꯕꯗꯣ ꯑꯗꯨꯅ ꯔꯤꯑꯦꯛꯁꯟꯁꯦ ꯍꯦꯟꯒꯠꯀꯗ꯭ꯔꯥ ꯍꯟꯊꯒꯗ꯭ꯔꯥ ꯍꯥꯏꯁꯦ ꯃꯥꯅ ꯗꯤꯁꯥꯏड ꯇꯧꯅꯤ ꯅꯪꯅ ꯑꯇꯣꯞꯄꯗꯣ ꯌꯥꯝ ꯍꯦꯟꯒꯠꯆꯕ ꯇꯥꯔꯒꯁꯨ ꯈ꯭ꯋꯥꯏꯗꯒꯤ ꯂꯤꯃꯤꯇꯦड ꯑꯣꯏꯅ ꯌꯥꯎꯔꯤꯕ ꯀ꯭ꯋꯥꯟꯇꯤꯇꯤꯗꯣ ꯑꯗꯨ ꯍꯦꯟꯒꯠꯇ꯭ꯔꯤꯉꯩꯗꯤ ꯐꯣꯇꯣꯁꯤꯟꯊꯦꯁꯤꯁꯀꯤ ꯔꯦꯠꯇ ꯍꯦꯟꯒꯠꯄ ꯂꯩꯔꯣꯏ ꯍꯥꯏꯅꯦꯕ. So the rate of any reaction will be decided by the factor which is present in the lowest quantity, lowest concentration. I will give you one example. Have you heard about relay race? amana chela amadong shwe amaduna amada so chen do khambra. For example, akhoi hum se lamjin chen dore lor se boje. Sir se am ti yang e. Yam e lor si ma so yam ti yang e ba. Ma di yam ya lamjin am slow e lor se. Adoi tardi lamjin se horen akhoi na winner oibo oida sa ayang ba ina decide to dina TV. Aina kan chela mon shwe ba. Ma no kan chela mon shwe ba. Man tap tam chela tardi yo roi. Khambra ina se hangi ko sir na se. Aduna horen winner oigadra oiroidra khwaidai yang buduna decide to roi. ꯈ꯭ꯋꯥꯏꯗꯒꯤ ꯅ ꯆꯦꯜꯂꯤꯗꯣ ꯃꯥ ꯌꯥꯡꯈꯠꯍꯟꯕ ꯑꯃ ꯗꯔꯀꯥꯔ ꯑꯣꯏꯌꯦꯕ ꯊꯣꯏꯒꯦ ꯍꯥꯏ ꯇꯥꯔꯗꯤ ꯑꯩꯈꯣꯏ ꯑꯅꯤꯗꯤ ꯍꯥꯟꯅꯗꯩ ꯑꯣꯜꯔꯦꯗꯤ ꯌꯥꯡꯖ ꯂꯩꯔꯦꯕ ꯌꯥꯔꯅꯤꯀꯣ so until unless we rectify the slowest quantity ꯗꯣ slowest factor ꯗꯨꯃ ꯍꯥꯟꯅ ꯁꯦꯝꯗꯣꯛꯇ꯭ꯔꯤꯉꯩꯗꯤ there will be no increase in the reaction rate ꯍꯦꯟꯒꯠꯂꯣꯏꯁꯤꯅꯤ ꯑꯗꯨꯅ any reaction ꯑꯃꯒꯤ ꯔꯦꯠꯁꯦ ꯀꯅꯥꯅ decide ꯇꯧꯅꯤꯕ ꯀꯔꯝꯕ ꯐꯦꯛꯇꯔꯅ ꯈ꯭ꯋꯥꯏꯗꯒꯤ limited ꯑꯣꯏꯅ ꯌꯥꯎꯔꯤꯕ ꯐꯦꯛꯇꯔꯗꯣ ꯑꯗꯨꯅ decide ꯇꯧꯅꯤ this is called law of limiting factor ꯍꯥꯏꯅ ꯀꯧꯋꯦ. Have you understood? ꯌꯥꯔꯦꯀꯣ so let me write here. So law of limiting factor states that kai haige ham tamda rate of a reaction noi NCERT gi do dum pabiu ko horen paba kan dadi polaung na budi tarni magi di maron khara sangdok samjin tawni ai hainadi swai swini ingsi rate of a reaction is decided by sin a law of limiting factor given by Sir Loyakawe is decided by the factor is decided by the factor which is present in lowest concentration, or I should say limited quantity. Limited oina yauriba factor do maduna decide tawniba. Adi sarna taiba reaction sida photosynthesis ki rate se hengal hange tare akhoi kana so hengal han batai. Carbon dioxide ki concentration se hengal han batai ba. Oi tardi hengalakadra hengal oidra hai basa. Carbon dioxide ki concentration do hongokpai matung ina takani hayak to sarna itus. Bro, yareko, nina, let's back to the factor again. So now I will start with ahanba factor is sir external da carbon dioxide han takip. Sir chandre pesam da taki ingat payadre so. Look here. Hozik sir na tak to ise carbon dioxide ki concentration na. Carbon dioxide ki concentration na photosynthesis ki rate ta kamaina affect toge isne. Yareko, Hamba yaubra yenge hosik present carbon dioxide concentration kayano in terms of percentage. Present carbon dioxide concentration say kayano haibakanda 0.03 to 0.04 ne. Yareko? Kya neba? 0.03 to how much? 0.04%. Yareko? Hosik present concentration say. Yareko? 하이리바 수마이나 레이람미바 짱세 시다기 까야보 행가토 마탐다 increase of carbon dioxide concentration from this normal concentration to 0.05 concentration bo hengatpa matamda yani there is overall increase in the rate of photosynthesis ki rate ke daweba hengat kaniba I'm going to write photosynthetic rate Okay, Tony, hang up the carbon dioxide hengalapuse. Carbon dioxide hengaladi global warming to ine. So it's good in some sense. When there is rise in the carbon dioxide concentration, the rate of photosynthesis increases. Hindi hengla. Yare ko. Adi hengalang gini hosik kana. Kay hengal hosik si carbon dioxide nam hengal pamatam ni. Natra. The world is developing. Carbon dioxide keep reducing. Carbon dioxide hengalai ni na hosik kay rate sukan tong gini. Ha na mamangay de tuamba photosynthesis ki rate de kitang di wanglap tong gini. Adubu, if we go beyond 0.05% it becomes damaging to the plant again, and after a while the plant will stop doing photosynthesis. Adu situation do la adi problem lay dunigo. Ana carbon dioxide am hengala ko phati. Hara henga pati aeva. Carbon dioxide ki concentration hengala ko tamda at first photosynthetic rate increases. This is called carbon dioxide fertilizing effect ko. Yarni ko asise. Adubu, if we go beyond, let me write, if we go beyond, kadai hena chaturaba, if we go beyond 0.05% sidagi hena lana hengalu tardi, it becomes damaging to the plant. Yani ko, fertilizing mo the poisoning effect when lakani. Kangi ko, it becomes damaging to plant. It becomes damaging over long period. Damaging to plant. Sima yaradro? Sina ahanba point naman note tawdonigo. Now, hosik sina percentage ta pibani, hosik present carbon dioxide concentration se. Kayano se ay bhapna sara harake, di, matamada koina 15-20 years ki mamangda tamgiba kandadi 380 ppm nen tame. Hosik te to suwa waro hengalamani. Compulsory hengat krabani hengalamani nati. Aduna hosik atmosphere sida lairiba carbon dioxide ki concentration se. Remember, it is averaged about 400 parts per million ppm ne. Micrometer the ps ko. Microliter. Hosik se kya neba? Carbon dioxide ki concentration se 400 ppm. Sina ppm the piba, ama sina percentage the piba unit to. Kangi ko percentage di kangi ko carbon di gas me amsi manungda carbon dioxide ki magi changse kya na no. So atmosphere is a mixture of the gas. Out of this mixture, nitrogen occupies 78%, right? Oxygen occupies 21%. Carbon dioxide is occupying 0.03 to 0.04%. High na habacity. Yare ko, sida pire sina ppm do ina pire. Si kai ki percent concentration eba present scenario. Hojik atmosphere sida lairiba carbon dioxide ki concentration si asini. Yare ko mayam si babada. Now, sida ina harak to say C3 plant ka C4 plants ka say. Let me start with C4 plants. C4 plants they have already got saturation at 360 ppm the saturation oil. C4 plants saturated at carbon dioxide concentration kya the saturated oil saturated at 360 ppm the saturated oil. Saturated edi khangi ko mabuk than do. For example, sir se nongmada yen maphai manga sanga melor se. Yare ko hojik ai ama chare ani pisale ahum sure kaya subda puk thandana manga subda saturated oil done. Yare ko adumain tawdana C4 plant se carbon dioxide concentration kya leba matamda mabuk thale haina 360 leba kanda hojik se present kya leire adri mana thanba changbe over the leire. So C4 plants ki oinadi the present carbon dioxide concentration is already saturated. Already saturated. Yare ko whereas C3 plants ki oinadi for C3 plants ki oinadi saturation se kadai waida uyeba saturation is seen only at 450 ppm the uyeb. Saturation is seen only at how much kai waida? Only at 450 parts per million concentration sida ufangeb. Yare ko C3 plants ki oinadi mabuk kya da thale hai na mabuk kya da thale hai no C3 ki oina C3 ki 450 da hojik leiba concentration sida kya adudi what ine hojik leiba concentration si is not what to say enough for C4 plants C3 plants for saturation. Wali ko about putting aside limiting hai kita adi the present carbon dioxide concentration is limiting for C3 plants ki oina di limiting oiri limiting adi low concentration da leiri hai ne yarni ko ꯑꯗꯨꯅ ꯁꯤꯗꯒꯤ ꯑꯩꯈꯣꯏꯅ ꯀꯩ ꯈꯪꯕ ꯉꯝꯃꯤ ꯁꯤꯃ ꯈꯪꯗ꯭ꯔꯗꯤ ꯁꯤꯅꯤꯁꯦ ꯈꯨꯝꯕ ꯌꯥꯔꯣꯏꯀꯣ ꯌꯥꯔꯅꯤꯀꯣ ꯑꯗꯨꯅ present carbon dioxide concentration से the present carbon dioxide concentration is limiting for C4 plants चुमिको चुमदेव is limiting for kanagi is limiting for C3 plants की limiting ओइरी हाइदो यारबो सिमा यारेको so this is all about the factor carbon dioxide की वारी है यारगे can I sense the slide now swipe up पामिनेगिस ꯀꯌꯥꯝ ꯍꯩꯔꯒꯦ ꯌꯦꯡꯒꯦꯕ ꯑꯩꯈꯣꯏ ꯃꯊꯪꯗ ꯔꯦꯁꯄꯤꯔꯦꯁꯟ ꯂꯩꯕ꯭ꯔꯣ ꯂꯩꯌꯦꯕ ꯔꯦꯁꯄꯤꯔꯦꯁꯟꯒ ꯀꯩꯒ ꯌꯥꯔꯦ ꯌꯥꯔꯦ ꯌꯦꯡꯉꯨꯔꯅꯨ ꯀꯣꯟ ꯇꯧꯔꯁꯤ ꯁꯣꯝꯗ ꯌꯦꯡꯉꯨ silence silence look here ꯁꯤ ꯆꯥꯔꯠꯁꯦ ꯍꯥꯟꯅ ꯄꯥꯃꯤꯟꯅꯁꯤꯀꯣ ꯅꯣꯏꯅ ꯑꯩꯉꯣꯟꯗ ꯍꯦꯟꯅ ꯍꯩ ꯂꯩꯔꯝꯃꯅꯤ ꯇꯥꯛꯄꯤꯔꯅꯨ ꯀꯥꯏꯅ ꯍꯥꯏꯔꯛꯀꯅꯤ ꯃꯔꯛꯇ ꯍꯣꯔꯦन ꯍꯩꯃꯟꯗꯅ look here let's do the comparison between C4 plants and C3 plants compare तꯧꯁꯦ ꯑꯩꯈꯣꯏꯗꯤ ꯁꯣꯝꯒꯤ ꯑꯣꯞꯁꯟꯁꯦ ꯑꯝꯇ ꯌꯦꯡꯉꯨꯅꯨꯀꯣ ꯑꯣꯞꯁꯟ ꯌꯥꯎꯗꯕꯤꯗ ꯈꯨꯝꯕ ꯍꯩꯕ꯭ꯔꯥ ꯌꯦꯡꯁꯦ cell type in which the Kelvin cycle takes place C3 the Kelvin cycle कायदा चथै mesophyll cell दा यानी को C4 plants the Kelvin cycle काय चथै मनुंगी inner cooler bundle seed cell लोइन चथै यारे को cell type in which the initial carboxylation reaction occurs carbon dioxide lotion basi C3 da kai loxali mesophyll cell da bundle seed cell da di mesophyll cell da solution male carbon dioxide initial hai bor male initial di ani makai da mesophyll cell da loi loxalan yari ko how many cell types does the leaf have that fix carbon dioxide carbon dioxide fix to bang ba cell type kya yoge C3 plant da only one mesophyll cell khatane yari ko whereas in C4 plant the carbon dioxide se mafam ani da fix away amana mesophyll cell da locally bundle seed cell da locally ba ana both mesophyll and bundle seed cell ani ko bundle seed da sungami mesophyll cell da sungami okay two yani ko bundle seed cell mesophyll cell ahan dishina kai one mesophyll cell khak se warni yare ko what is the primary carbon dioxide acceptor in C3 plants RUBP very good how about this one phosphoenol pyruvate yare ko number of carbons in the primary carbon dioxide acceptor in C3 plants 5 carbon and this is 3 carbon what is the primary carbon dioxide fixation product here it is 3-phosphoglyceric acid. Here we have oxaloacetic acid. Hangi. Number of carbon in the primary carbon dioxide fixation product. This is a 3-carbon compound. This is a 4-carbon compound. Yaraba? Does the plants have rubisco? Answer is yes. How about this? Yes. Yes, yawon budi kenai. Amana mesophyll cell da, amana bundle seed cell. Does the plants have peptides? Here, no. How about in C4 plant? Yes. Which cells in the plants have rubisco? Answer will be mesophyll cell. Rubisco ka yaudano? Bundle seed cell manungda yaudane. Ready ko? Carbon dioxide fixation rate under high light condition. Light yam yaksan tari carbon dioxide fixation rate under granny low siladi handaba layroi because it can avoid photorespiration. Highlight intensity da kai thok no. Siladi photorespiration thokadro. Thoroidamna the rate remain high. Adum high oiler ni haine. Yareko, how about here? Whether photorespiration is present at low light intensity? No. Photorespiration, the highlight intensity the thokadam natro, so animakta am toy low light. Yareko, whether photorespiration present at high light intensity? Yes here, no here, because it has got adaptation to avoid it. Right? Next, high low. I should say negligible for this one. Yareko, next, whether photorespiration would be present at low carbon dioxide concentration? Carbon dioxide concentration low body kanana wangi haina. Okay, will there be here? Yes. How about here? No. Let's not say no. Negligible. ꯌꯥꯔꯦꯀꯣ ꯀꯩꯒꯤꯅꯣ ꯊꯣꯛꯇ꯭ꯔꯤꯗꯤ adaptationꯗꯣ Kranz anatomy ꯒ dimorphic chloroplast ꯒꯗꯨꯅ whether photorespiration would be present at high carbon dioxide concentration never ꯁꯨꯡꯊꯣꯛ ꯊꯣꯛꯑꯔꯣꯏ carbon dioxide ꯌꯥꯝ ꯋꯥꯡꯉꯤꯉꯩꯗꯗꯤ ꯀꯩꯗꯧ ꯅꯨꯡꯊꯣꯛꯏꯀꯣ ꯌꯥꯔꯦꯀꯣ temperature optimum C3 plants ꯅ ꯈ꯭ꯋꯥꯏꯗꯒꯤ ꯄꯥꯝꯖꯕ optimum temperature ꯁꯤ is about 20 to 25 degrees Celsius ꯑꯃꯁꯤꯗꯤ ꯑꯁꯥꯕꯗ ꯍꯩꯅꯔꯣꯏꯗ꯭ꯔꯥ above 40 sorry 30 to 40 degrees Celsius for C4 plants there. May I know few examples of C3 plants? Nang uri sing se naman. The mango, the banyan, these are all C3 plants. Yarni ko, C4 plants, I have given three examples. Maize, sugarcane, and sorghum. Lower is Yari ko. Now look here. Next. Ay ngasari sir na ngangi ma'am do si ma'am so lo yaurabani ko. Amtang paraga yung birasit. Loy mag yaurabani. Now let's talk about light. Kitang kap palamige. Ay morning dadi light se pa kitrabani. Sida ꯑꯗꯨꯝ ꯍꯥꯏꯔꯛꯑꯒꯦꯀꯣ ꯄꯥꯔ ꯌꯦꯡꯕꯤꯔꯨꯔꯣ ꯌꯥꯝ ꯂꯨꯗ꯭ꯔꯦꯕ ꯌꯥꯝꯅ ꯑꯗꯨꯅꯅꯦ ꯑꯩꯅ ꯍꯥꯏꯔꯛꯏꯁꯤ ꯂꯥꯏꯠ ꯏꯟꯇꯦꯟꯁꯤꯇꯤꯁꯦ ꯁꯨꯝ ꯍꯦꯟꯒꯠꯂꯦ ꯍꯥꯏꯗꯤ ꯑꯌꯨꯛ ꯃꯝꯅꯤꯕꯀꯣ ꯅꯨꯡꯁꯥꯁꯦ ꯇꯞꯅ ꯇꯞꯅ ꯈꯔ ꯁꯥꯗꯣꯛꯄ ꯍꯧꯔꯦ ꯈꯔ ꯁꯥꯗꯣꯛꯄ ꯍꯧꯔꯦ ꯍꯥꯏꯗꯤ ꯐꯣꯇꯣꯁꯤꯟꯊꯦꯁꯤꯁ ꯔꯦꯠ ꯍꯦꯟꯒꯠꯂꯅꯤꯀꯣ ꯍꯦꯟꯒꯠꯂꯅꯤ ꯍꯣꯔꯦꯟ ꯅꯨꯡꯁꯥ ꯌꯥꯝ ꯁꯥꯒꯠꯂꯀꯥꯟꯗꯗꯤ ꯐꯣꯇꯣꯁꯤꯟꯊꯦꯁꯤꯁ ꯔꯦꯠꯁꯦ ꯑꯃꯨꯛ ꯍꯦꯟꯒꯠꯂꯨ ꯌꯥꯗꯦꯕ ꯀꯣꯟꯁꯇꯦꯟ ꯑꯣꯏꯅ ꯆꯠꯈꯤꯅꯦꯕ ꯈꯪꯕ꯭ꯔꯣ ꯁꯔꯅ ꯍꯥꯏꯁꯦ तो अदना rate of photosynthesis का light का कई मरी लेने के है ताड़ी when the light intensity increases photosynthesis rate also increases but अदुम एंगल ले गजरा दी लई रोएगा यानी को constant होने लेगिनी असीमा का खंबी बता रहे यारे को now ngasai aina carbon dioxide की इग्रिदो में सीधे इरे temperature दको हम तंग ngai go temperature da temperature hengat pa matam da which one respond better to temperature C4 plants. Yarnika, C3 plants, the temperature kai hen hengatu tardi, they may face the problem of photorespiration as a reason the rate decline. Harake hen hengaluradi animak impact lakanika due to denaturation of enzyme for both the cases. Yarnika, water gi ama horen yauriba. Water gi role sidi direct nati indirect nina. Position of C lines important. Direct nataba indirect adi ising am pangdadi quirani pambido. The plant will be wilted. Hidata adi stomata gadom to open to yaroy. Huizalla ikradi. Stomata open to the changaroy. Carbon dioxide gases, there will be no gases exchange. Without carbon dioxide coming in, there will be no photosynthesis. Anika, please go through the other factors. Yarabo sivokta, sivotor sigo. Thank you.